Hello and welcome to a brand new episode of KU Pulse, the podcast. My name is Hisham Akhtar, part of Khalifa University Enterprises Company. Our topic today lies at the intersection of science, biology, health, and imagination. Gene editing, lab-grown organs, and even the science of de-extinction. It sounds like the plot to the next Hollywood movie, but it's reality and it's happening right now. To make sense of it all, I am delighted to welcome Dr. Shabir Hassan, who is Assistant Professor in the Biological Sciences Department at Khalifa University. His career spans everything that we will talk about today, so welcome to the show.
Thank you very much, Hisham, and glad to be here on KU Podcast.
Great. So before we get into the technicalities of the topic today, what's your story? How did you end up on this podcast? What made you interested in the science to begin with?
I come from Kashmir. I did my bachelor's there in chemistry, biology, and English. Then I followed it, trying to actually get into biotechnology because biotechnology was like on the horizon. It was a new thing in the town. Got really excited to do that, and then followed my passion also for physics, for which I did my PhD in physics. Moved to Switzerland, and from there, I wanted to actually get into something where I can have a hands-on experience in translational as well. So I want to work on something where you will have a product at the end of the day, which can actually benefit the humanity in broader terms. And then, because the work I was doing during my PhD, I was literally looking at how proteins communicate and talk to each other at a very molecular and atomic scale, looking at 10 to the power minus 12 or 10 to the power minus 15 seconds, the femtosecond spectroscopy or like nanosecond spectroscopy. But had I continued in that one, it would not mean that I would be able to start my own lab. And since the passion of being able to create inventions and innovations that could actually benefit the general population, I moved to the US. I was at Harvard and MIT, wherein I worked on biomaterials and creating tissues and organs in the lab to be able to answer questions on, for example, organ donations and injuries, or people when they lose limbs, people when they lose tissues, and this can have effect on people who are suffering from, for example, diabetes, diabetic foot, burn wound injuries, in myocardial infarction, or even people in the battlefield. And slowly, the interest actually got me into developing those kind of technologies, which will help us build tissues and organs in the future. And I know that KU Pulse has been doing a great job, bringing to public the inventions or innovations that are happening at Khalifa University. I think that's why I'm here to talk about what is actually happening in my lab and what could happen in my lab in the future.
Great. Absolutely. We talk about all things STEM on KU Pulse. So you've been all around the world. Clearly, you've been doing this for a long time. So let's set the big picture for the listeners. And why now? Why are we able to talk about lab-grown organs, de-extinction, gene editing? Are there any significant breakthroughs or technological advances that are helping transition all of this from theory to reality?
That's a very wonderful question. The thing is that people generally think things have been blooming out all of a sudden, and then you see everything on the horizon, and then you believe that it actually happened yesterday. But honestly, it's not just now. It has been happening over the last 3-4 decades. If any of us could actually remember that we had those kind of news items coming into the different multimedia forums, saying that scientists are this close to printing an organ in the lab. This has been happening for the last 30 years. But are we there yet? The answer is no. It's only through social media and other outlets or other forums, other platforms, where people have been more sensitized about what's actually happening in the scientific world, and they have shown that kind of an interest. That's why we see that now these inventions and innovations are actually getting out, and people are trying to know about that. But having said that, there is always a time frame wherein you have inventions and innovations going at a very slow pace, but then all of a sudden there is some kind of invention that actually fast-paces everything. And that has been happening in the last, I would say, a decade. We had different types of technologies wherein people were already trying to do gene editing, trying to actually correct some gene which was not functioning properly, or if a person was born with an additional pair of a gene or a gene which was not in its correct form, so it could actually lead to malfunctions. And people did use different types of platforms to be able to correct that particular malfunction. It didn't work that way as it was supposed to be. And then came a technology, for example, CRISPR-Cas9, which is a molecular scissor that helps you cut down any DNA or insert anything new in the DNA in a very precise manner. And while people are trying to know more about CRISPR-Cas9, only last month there was one more technology. It's called a single-strand nuclease that got published in Nature.com, when you have a technology which might actually even be at the same level or even better for making additions or editing in a single strand DNA, which was not there. But I mean, we will be able to know more about that technology once it goes in the mainstream scientific world. For now, I think people are trying to actually really understand what gene editing is, what it can do and what it can't do. And then you also have regulations to be able to put a hold on what it can do and what it should not do. So those things are definitely there, but I think it's more about the information being put forward on different platforms for the general public that they got interested in. That's why you see a lot of buzz right now happening around these kind of technologies. And then recently, like I think 3-4 years ago, there was a Nobel Prize in 2020 for the CRISPR-Cas9 technology that was given to two ladies. Pretty brilliant work, and we might see more recognitions in the future. For example, this year's breakthrough prize was given to David Liu, actually for even fine-tuning the CRISPR-Cas9 to a single nucleotide resolution. That means that even though you knew that there was something wrong, for example, with this arm of my coat, and then CRISPR would actually say, oh, by the way, these many centimeters are defective. I will try to actually perform some restoration in this area. While as what David actually is doing, for example, David Liu from Harvard, it is literally telling you which stitch needs to be removed and which stitch needs to be repaired. So that you generally have less of malfunctioning afterwards, because you might be able to create new organisms with just CRISPR-Cas9 or any other gene editing technology if it is not performed precisely. Because any addition or deletion in the genome will result in the formation of an organism which is deviant from what it should be naturally. So automatically you might be talking about a new organism altogether.
Very, very interesting. I mean, clearly a lot is happening and has happened over the past 20, 30 years.
Yeah.
So maybe let's unpack that a little bit, right? I'm sure our listeners, when they hear about lab-grown organs, they're like, how does one even go about doing that, right? So maybe you can walk us through that process a little bit.
Yeah.
And, I'm sure the question on everyone's minds is, can they actually function like the real organs that are there? We hear about even during organ donation and transplants, the body naturally might reject certain organs, right? So talk to us about that a little bit.
I think it's a very loaded question. It has two parts. One part is whether or not it will function exactly the way it is supposed to function in human biology, in human body. The answer is maybe yes. Better than what we used to have. So if you, for example, look at how people studied different diseases, pathophysiology, how diseases actually, work, what are the different things that are activated, what are the different types of proteins that are released, how does a drug affect any disease, for example, cancer. So the go-to technology was using cell lines, like there is a cancer cell line, or there is a cancer cell line primarily taken from the organism or from the human. You could directly take that cell line, purify it, and then do an experiment directly on the cell line. Or you could immortalize the cell line, then you can have different types of populations of these cells over a prolonged period of time, and then you could do many other experiments. But one of the key aspects to this growing of cells in the lab is that they're all grown in a single layer. We call them monolayers. But so it's two-dimensional. But we know that humans are three-dimensional.
Yeah.
So why did we go wrong? You can't expect a two-dimensional monolayer perform or give you the results exactly the way it is happening in humans. Because I can give an example. It's a very funny example. People are saying, oh, by the way, this thing and that thing is anti-cancerous. Now, if you have this tea, it will actually treat cancer. If you have now curcumin, it will treat cancer. If you eat maybe a burger as well.
Yeah.
Why it is funny is that even if you spit, like just normal spit, in a 2D monolayer of cells, it is going to kill the cells. Whether it was the spit that did the miracle, or it was the bacteria that were present in your mouth that actually killed the cells. But eventually you had the cells dead.
Right.
And if we look at roughly the take-home message, you killed cancer cells with your spit.
Yeah, and then, the grand headlines on social media come out and all of these things.
But is that right?
Yeah.
No, because our bodies function differently. And that's where this three-dimensional or four-dimensional tissue engineering comes into play, where we are making artificial tissues in the lab. And what we do is that we actually put in the hierarchical structural complexity into this monolayer. What we do is that we go for three-dimensional from a two-dimensional thing by way of adding different materials, which could actually also add a mechanical component to it. Because we know that, if we, for example, look at our hand, our organs, and if there is any monster that comes and then he or she or it, whatever, they actually cut my arm, for example. I give this example a lot. Now, what do you see from this point? In the middle, you see, for example, bone marrow, if it is humerus. Then you see the bone. Then you see muscles. Then you see maybe, blood vessels. Then you see skin. So there's a lot of heterogeneity. It goes from the bone marrow all the way to the outer skin layer, right? Now we know that the bones are very tough. They are not like skin. And skin is unlike what meat is, muscle, right? And muscles are different than blood vessels. So you can't have one size fits all to all this heterogeneity. Now, if you need to make something which will recapitulate physiology or pathophysiology in its realistic sense, you also have to make a tissue which is mechanically similar to what we are trying to treat. If, for example, I was working with bone tissue engineering, we were trying to actually see if we could make artificial tissues in the lab that will help us repair a broken bone. For that, we took a different material which had the same mechanical strength as you find it in the natural bone. And in the other project, when we were trying to look at whether or not we can use a platform to heal a myocardial infarct, that is a heart attack. So during a heart attack, there is a damage to a certain part of the heart. And then that part of the heart is dead. There are dead cells. There is no oxygen in there. But we needed something, because we know that heart is a very active organ than a bone. It's always in the motion. So we wanted to have some material which has a lot of tensile strength. For that, we used silk-based biomaterial. And because silk also has a natural tendency that you can stretch it without breaking it. And we could show that, the mechanical match is very important. Whatever you're doing in the lab, it has to actually match mechanically to the tissue that you are targeting. Then comes the point whether or not it will work or not. The answer, so I gave you a perspective of three-dimensional, like the dimensionality of the human tissues to the tissues that we are trying to actually make, or the models in the lab. So that means that from the mechanical component of it, we already are taking care of this. But what is also interesting that takes it one step further to the personalized medicine regime is you could now have cells in this three-dimensional matrix that you have to apply on a patient to repair a bone, skin, eye, ear, whatever. You talked about rejection, immune rejection. And immune rejection generally happens when the body thinks that it is being attacked, or anything that's introduced in the body does not belong to it. Then the immune system gets activated and you eventually get a failure. In this situation, what we could do is we take the cells from the patient himself or herself, and then we take out certain cell types which we believe can actually differentiate further. And then with particular chemokines or other chemicals, biological entities, we make these cells, which are stem cells, differentiate into the type of tissue we want in that patient. So the patient actually gets treated by the cells that are derived from the patient himself or herself. So that actually lowers the immune response from the body. That means that we will have a higher chance of success rate of this fabricated tissue that is fabricated in the lab to be accepted by the patient. That is one aspect. The other aspect is something called as intergenome variability. What does that mean? That means that Hisham is here, Shabir is here. We both are humans. 99, maybe 100% genome is there, but we do have certain differences, very subtle differences in our genome. That makes you you, and that makes me, right? It's not just like you have a beard, I don't have, but it's also the personalities. It's also, for example, the height, the shape of the nose, or, for example, how do you look, your height, what are the different factors that can actually affect you. But at the same time, if I want to, for example, explain it to general public, imagine at 0000 hours, you also got a headache, I also got a headache. Both of us take Panadol, or for that matter, any paracetamol, right? I can guarantee you that there is never a chance, maybe 1 in a trillion chance, that we, both of us, will get relieved exactly at the same time from the headache. That's never going to happen. Why? Because the bodies act differently. Our bodies are different. And that's why if you, for example, look at twins, there are twins who are born together. They share the womb together for 9 months. But even if there is one twin, one of the twins, who actually has any organ failure, and the other twin gives him or her his or her own organ, there is still a possibility that there might be immune rejection. Why? The bodies are extremely different. They actually say that we don't want to touch it. So that is where this thing comes in, that you might have a success by creating models in the lab which recapitulate human physiology more than anything else. But at the same time, you might also want to, for example, create things which are faster and which are actually into an actually active physiological system. For that, you create these avatars in small animals, wherein the animal also has the same kind of disease that a human has. And then you have humanized forms of these animals. And that's where this whole debate comes in, that can you now actually also grow organs in animals that are humanized? And we saw an example of hearts being made in pigs, but these pigs are not just the general pigs that you see in different countries. These pigs are also genetically engineered to a level wherein they perform more like a human being from a biological perspective, and then there are also certain deletions in the genome which make them or any of their organs to be not rejected by the humans, even though we are not actually getting a lot of success there. But I think scientists are working still in that direction. Maybe in the next few years, we might have something, a breakthrough that might help people to cut short this organ donation or organ shortage problem that we actually face all over the world.
Interesting. And I mean, the three-dimensionality aspect is solved. Are we right to say that CRISPR and gene editing is helping potentially increase the success rates of whatever is transplanted into human beings by personalizing a little bit what is being developed in the lab at this stage?
I think it is a complex question in the sense that you don't necessarily need CRISPR-Cas9 to make, for example, engineered tissues in the lab. You might need CRISPR to make bigger organs, or you might need CRISPR or any other gene editing technology to be able to grow humanized organs. But if we're talking about, for example, making the tissues and organs in the lab, you might not necessarily need CRISPR in the first place. What you might need CRISPR, though, is actually also, like, for example, taking a cell line, which is from the patient, and then in the lab, fixing it using CRISPR-Cas9 or not just Cas9, but any other Cas as well, or any other gene editing tool, and then putting it back into the patient so that now it actually gets divides further, and then it takes over the diseased organ or tissue. But if you are only focusing on organs and tissues being made in the lab, you necessarily have to focus on the scaffold that I was talking about. That's the extra, we call it extracellular matrix. That is kind of the highway that you have to build on which the cells, which are like, the cars and trucks, which have to move so that there's better communication between the implanted tissue or organ and the host animal. But CRISPR-Cas9 has a role in raising the humanized organs in different animals, but at the same time, also maybe editing any kind of diseased cell or tissue in the lab first, and then reintroducing it in the patient to be able to correct it there in a more physiological sense.
Okay. On the topic of gene editing, so like you mentioned, disease cells and things like that can be improved. How far can we take gene editing? I'm sure people are wondering, is Captain America going to be a reality soon, right? Super serum, anti-aging, because there's a lot of debates and discussions, about follow this diet, follow that diet. But is there, a possibility, maybe not today, but in the future, where gene editing is a shortcut in a way to these things?
I think people, for example, in our lab, we are now moving, not just moving, but we are also complementing our 3D tissue cultures with something called as avatars. So we have a small animal model. For example, we have the zebrafish we are taking. And we're trying to make avatars in zebrafish that resemble an actual patient in the hospital. So we are, for now, for example, focusing on multiple sclerosis or any other neurodegenerative disease or any other disease that might actually be, affecting humans quite a lot. So what we do is that we try to, first of all, see whether or not there is any homolog, that's whether there is a similar gene in the zebrafish that has the same action that it has in humans. And then if that particular gene is affected in humans, we need to see if there is a match in the zebrafish. Then we can try to edit that in the zebrafish to be able to make a one-to-one copy of the patient in the zebrafish. And this word is now actually used quite regularly in avatars. We call them avatars because it comes from the movie Avatar, wherein you are making an animal avatar of a human patient. And there is a possibility that you might actually know more about the disease, or you might understand pathophysiology better in these avatars. But what we are trying to do in our lab is that once we have this avatar, which is similar to what people are actually in the US or other places are doing by recreating or trying to reintroduce different animals which are extinct, is what we want to do is that we want to make this new. It's actually a new animal. It's not an avatar, but this animal we will try to utilize for understanding the disease better, but at the same time maybe repurposing some of the already FDA-approved drugs which are available in the market. So that our goal will be to then correlate it one-to-one with whatever we get from the disease models or the avatars, and we try to actually make it one-to-one in the 3D tissue culture model, and we try to see whether we have a hit of any drug, and we'll actually go about that. Now, taking it one step further, not using it as a model for diseases, you could actually also say, by the way, we are making a new species of fish, which is true. But then if you introduce it in the public as making a new species of fish, it will catch more eyeballs than if you're saying, by the way, we want to understand this. And that's precisely what, for example, the companies like Colossal have done. They started a project to revive the woolly mammoth. Why? Because they give their own reasons. They want to see if it can actually have any kind of ecological or environmental balance again, But then in the process of that one, because we know that, it's not going to be easy, you need to first of all see how you have a DNA. So there is also there are like a couple of processes. There are in total 3 different processes that you can bring any animal back. One is, we all know, it's called cloning. So people actually do gene cloning, and we had the curious case of, we had llamas, we had also camels here in this part of the world. But we had the Dolly the sheep, the first one. The other one is called backbreeding. So you, for example, have certain characteristics in one or a few members of the population that was present in something that was already existing before. And that can be then backbred. You are actually breeding it with something which also has similar kind of traits. And then until you try to actually increase the traits of that particular gene, which we're looking at. Then the third one is what Colossal did. You have the DNA from fossils or anywhere else, and you try to see what is it that makes it different, and whether or not we have any animal relative from an evolutionary perspective. And an example is of the dire wolf that they introduced very recently, I think last month, which they had already made 6 months ago. So what they did is that they did it because of only the attention. Most of the people who are actually watching me also have, watched different types of. They have maybe played different types of games, or watched Game of Thrones, or read any other book. And what you see is these beautiful giant creatures, which are like wolves, white in color, and they are also like very dreadful, but at the same time pretty beautiful as well. And what they did is that the Colossal Biosciences, they saw that the dire wolf, the remains that they got from the fossils, dire wolf, the next relative to dire wolf in today's world is actually the gray wolf. It is 99.5% of the same genome. So the only thing that actually makes dire wolf is 0.5% of the DNA that was present in dire wolf. But what actually did happen is that you took the DNA from the these specimens that they saw, and then they would then match it with the gray wolf that's already present, right? And with the 99.5% match, they looked at the 0.5%. That makes dire wolf. And dire wolf, they are not actually these giant animals. They're only 25% bigger than an actual wolf. But then they were, shown the way in the folklore or in different books and TV series that they are kind of very giant. So out of this 0.5% DNA, the Colossal Biosciences, what they did is that they focused on some of the traits that will make it look exactly like a dire wolf, different than the gray wolf. So they worked on, for example, the size, the gene that affects the size, the gene that affects the fur color and fur size. And then they used another platform wherein they made certain changes there, and then they took the egg from the wolf, and then tried to actually modify it with the CRISPR-Cas9 to be able to introduce these new traits that are present in the dire wolf, and then impregnated it into the dog species. And then after that, they got like 3 dire wolves. 2 are males and 1 is female. But are they dire wolves? And did they create dire wolves? The blunt answer and brute answer is no. Right. They are not dire wolves. They are actually gray wolves, which look like dire wolves from their appearance. Because if you look at them from a biological perspective, the 3 dire wolves that they made, 2 are male and 1 is a female, but they are actually clones of each other. So they should behave exactly how, like all the 3 should behave exactly the same. But we see that the two males, 1 is very not aggressive, but very playful. He's very, I would say, he's very active. Yeah. But the other one is pretty shy, because there are a lot of factors that actually affect what becomes of you. Environment plays a huge role. The kind of interactions you have, the way you were born, the mother you were born from, the surrogate mother that the female dog is there, right? So all these factors will come in, and I would not be hesitant in saying that they made something which looks like a dire wolf, but molecularly or from a genetic perspective, it is not a dire wolf. Mm-hmm. It seemingly looks like a dire wolf.
Setting aside, the sensationalism around woolly mammoths and de-extinction and everything. What do you see as the actual scientific benefit of something like this? Like, because I mean, they're pursuing this project or this research for a broader end goal, right? So de-extinction research or science, or let's say, the ability to at least visually recreate animals that may have gone extinct. What do you see that as the benefit for the average person?
I think every person will not benefit from this, though. But if we're looking at the oral effect it might have on the diversity, we know that we have several reports that suggest that in the next 25 years, that's by 2050, if we continue to grow the way we are growing, and if there is nothing that needs to be done from the perspective of preservation of species, then 50% of these Earth species, be it animals, plants, bacteria, fungi, they'll go extinct in 25 years. So when we look at this de-extinction project, I think the biggest takeaway is that we might be able to save the species which are about or on the brink of extinction, to bring them back. But however sad it might sound or ironic it might sound, is that Colossal Biosciences has strictly said they are not letting these direwolves, which they presume are direwolves, not to interbreed. And that means that in 20 years, you are going to make the direwolves extinct again. Okay. I hope that does not happen. I hope that the science, the beauty of science that comes in, and the reasoning for that, the justification is that we already have the endangered species list. We better, for example, work on those species which are about to go extinct, and then try to utilize the technologies that we have, that we learned from development of the oral bringing in of woolly mouse or dire wolf, or maybe woolly mammoth in the future, and utilize this technology for saving any of the species that we believe are on the endangered list and might go extinct. Right. That is the biggest takeaway as a scientist and as a human of this earth that I see in this technology.
So rebalancing ecosystems.
Absolutely.
And the environment.
Absolutely.
And I mean, we've been talking about the applications within animals and species, but can this be applied to marine life as well? So let's say preserving coral reefs and things like that, because as we know, majority of the planet is water, and there are huge efforts to preserve aquatic life. So
I think absolutely. Honestly, it will be. It can be applied to anything or any other species which we believe exists here, because at least we know what environment it exists in, what is the climate that it needs, what does it actually feed on. So if we have all that data, it might make our lives a lot easier to be able to see or have more success rate of any other project that we might be doing for example, for saving any other species, be it from coral reef side or from plants, or even the extremophiles that we find in deserts and other areas. And not just the deserts, but if we talk about the UAE as well, we are blessed with an abundant number of species which are very exotic to our area. And then if there is a species which we believe might go extinct, because I was reading very recently that there was this desert cat which was actually photographed for the first time after so many years. Maybe it is actually on the verge of or it is an endangered classified as an endangered species. Maybe we might be able to use that technology to be able to save these species for the overall betterment of our area as well as for the earth. So I believe that the oral applications are endless, and it doesn't necessarily have to be applied only to animals, but it can be applied to any other living species which is still living and might not go extinct in the 25 years.
Great. I mean, lots of positive things happening. What excites you about the next 10 years, let's say, in biotechnology? Both, I would say, as a scientist, of course, but also in terms of the possibilities and that aspect of imagination coming into reality. So what should we be looking forward to?
I think excitement is
Do scientists get excited?
They do, but I mean, it's also excitement can be both positive excitement as well as negative. So I'm positively excited about the possibility of having lab-grown organs, which can actually replace the organs shortage that we already have, or the organ donation demand that we have in the world. That I see is a possibility in the next 10 years. But what also actually my fears in the next 10 years are that we are literally very close to introducing new species in the world order. So that means that even though we might be playing, and trying to actually bring something back or create or fix something, but in this process, we need to make sure that we are actually using the tools which are already regulated and but which are also monitored from the perspective of not going in the wrong hands to be able to create what they want to create. But at the same time, I think there's also this, there's always this balance that you don't necessarily create what you think you created, because epigenetics or other factors actually also weigh in, and they do play a huge role in that aspect as well.
Yeah, I think that question of just because we can, should we, and we've seen examples of that with the internet, AI now, there's a lot of debates around that. So any final words for our listeners around this topic? Any interesting research papers that you're working on at the moment?
Right now, we are trying to, honestly, I mean, from I can talk about it from my perspective. We are trying to actually look, work on the diseases which are hard to treat. For example, we have some projects running on spinal cord injuries. We have projects running on multiple sclerosis. There's ovarian cancer, and we are also trying to get into this endometriosis and trying to find out if there's any possibility of make a new model to be able to understand the disease better, and then we might be able to treat. The other project which I'm really, really excited about is called chronic traumatic encephalopathy, CTE. Unfortunately, until now, people have been diagnosed only post-mortem. That's people have to die to be diagnosed for that particular disease. But we are moving towards making a new scale wherein we might be able to diagnose the patient before he or she dies. That's one thing. The other thing that I'm personally very excited about is also this avatars, making avatars in zebrafish and trying to actually utilize different omics approaches. We're fortunate to have the support from the Biotechnology Center. We believe that we have the necessary equipment and the facilities here at Khalifa University that might be able to answer or even find us novel targets for repurposing of any other drug. We have some candidates, and I'm very excited about looking at whether or not there is already an FDA drug which is approved for something else. If we can repurpose that and try to actually answer the burning question on multiple sclerosis or neurodegenerative diseases, including ALS and other stuff, and that we have also things running on NAFL, non-alcoholic fatty liver disease, and chronic traumatic encephalopathy, as I already said. That's what I'm actually very excited about, and I have a huge faith on the gene editing technology to be able to give us more realistic pathophysiological models so that we can understand disease better. And then once we have the proper knowledge of understanding a disease, we might be able to also come up with a novel solution that will work in humans better than it has been so far.
Seems like we might have to do a follow-up episode for the podcast in the future.
Absolutely.
But thank you so much for joining us today. Insightful conversation, lots of positives, potentials. There might be a future where we can have printed organs, from vending machines at some point.
Or designer meat or designer animals.
Yeah, designer animals. Once we get into customization. But that's where the imagination part comes in. To our listeners, if you enjoyed the episode, please subscribe to KU Pulse, the podcast on YouTube, Spotify, and SoundCloud. Thank you so much for your time today. And until next time, stay curious.
Thanks a lot for the invitation. And as you said, stay mad and stay curious. Thank you.
Thank you.